Hi, my name is Tim Sheahan and I study viruses that make people sick. Today I want to share an exciting story about hepatitis C virus, or HCV, that I developed while a postdoctoral fellow in Charlie Rice's lab at the Rockefeller University. 70% of the people that get infected with HCV are chronically infected, and over time this can lead to the development of liver cancer. Interestingly, 30% of those infected can fight off the virus completely. In 2009, it was discovered that mutations in the interferon lambda 3 gene were associated with the outcome of HCV infection. People with the CC mutation, shown in blue, were more likely to successfully fight the virus. In contrast, people with the TT mutation, shown in red, were more likely to develop chronic infection. So in the lab, we really wanted to figure out what might be guiding this clinical dichotomy. We wanted to distill the complex interactions in the patient down to the singular interaction between HCV and the target cell in the liver, the hepatocyte. If we could define how this cell is reacting, perhaps this would inform us as to the molecular mechanisms guiding the interferon lambda 3 associated outcomes in the clinic. We were faced with a problem. Hepatocytes are challenging to use in the lab. It's hard to mark cells as infected and only a small subset of cells get infected. But luckily, a very talented postdoc in the Rice lab, Chris Jones, developed a cellular reporter for HCV that made it possible to view infection in real time. Without going into too much detail, uninfected cells have a red fluorescent protein in the cytoplasm, and upon infection with HCV, the red fluorescent protein is relocalized to the nucleus. Thus, the readout is binary, where infected cells have red nuclei and non-infected cells have red cytoplasm. What I want to show you now is a black and white movie I took of HCV infection of primary human hepatocytes. The movie starts 12 hours after infection has begun. Keep an eye on the cell that I've circled in orange on the right. Watch as the donut-like structure of reporter protein is relocalized to the nucleus upon infection. And now I'll show it again. So now we had a means of marking cells as infected. We just needed to figure out a way to isolate the infected cells away from the majority of cells that were not infected. The rationale for isolating only infected cells is that if we were to study the cells as a population, the signal from non-infected cells would likely muddle the signal from infected cells, the cells that we're really interested in. To isolate infected cells away from non-infected cells, we decided to use a really cool technique called laser capture microdissection. This technique is driven by the affinity of two materials, the silicone in the collection tube cap and the slide membrane that your sample would be on. And the silicone and the membrane are sticky for each other. You can even hear it as they stick. The LCM Rig at the Rockefeller University is an inverted microscope fitted with a robotic stage controlled by this touchscreen. I'm pointing to hepatocytes that were grown and infected on the slide membrane and then fixed. The LCM slide is braced by a glass slide to provide some rigidity to the membrane, and then this sandwich is put into the robotic stage. So if you look at it from the side, the fixed cells are facing the glass, and the clean side of the membrane is facing up. The collection tube cap, as you can see, is filled with a silicone plug. The collection tube is fit into this holder, and then that's put into the robotic stage. The slide can move in the X and Y dimension, and moving the tube in the Z dimension is what facilitates capture. Multiple cells or groups of cells can be captured at a time. In this example, we capture Rice lab from a monolayer of cells. The machine will then cut each letter out sequentially. Upon completion, you lift the collection cap tube up, and just to make sure we've collected Rice lab, we can inspect the cap surface. And there we have Rice lab. Coupling this technology with the cellular reporter, we can now isolate HCV infected cells. Here we have in the middle of this field a group of cells with red nuclei, so they are HCV infected. The machine goes and cuts out that group of cells. You lift up the cap to collect them, and just to make sure that we've collected those infected cells, we can look at the cap tube surface, and here we have that cluster. So for each cell or population of interest, we just grab a new tube and put the others on ice until downstream processing. Using this technology, we isolated HCV-infected cells or cells next to infected cells and analyzed their transcriptomes. We did this using cells from donors with different interferon lambda 3 genotypes. This was a ton of work and yielded lots of cool results, but one of the most interesting was that infection frequency was increased in cells with clinically unfavorable interferon lambda 3 genotypes. Another cool finding was that cells with favorable interferon lambda 3 genotypes had better antiviral responses. In summary, our experiments provided insight into the molecular mechanisms guiding the outcome of infection in the target cell of HCV and provided an explanation for the clinical dichotomy seen in patients. These results could not have been generated without the ability to isolate infected and adjacent cells via LCM. Learning this technique was a lot of fun, and I'm happy to be able to share this process with you in this video. My hope is that other people will be inspired to apply this technique to their own work, leading to discoveries that we couldn't possibly have predicted. Thanks.